Hello, my name is Francisca Rodríguez-Cabello, Visual Student Researcher at the Center for Biotechnology and Bioengineering at the Universidad de Chile. Today I will present the project called Design of Peptides with Desirable Activity assisted by Machine Learning and Data Mining Strategies. To start, peptides are protein structure composed mainly of amino acids linked together by a peptide bond. They have different properties and characteristics that make them unique and provide various advantages in industrial, pharmacological, and biotechnological applications. One of the main problems of working with this type of structures lies in the high economic cost involved in its synthesis. Therefore, it is necessary to design and implement computational methods that facilitate the design of peptides with desirable activity. Based on an analytical approach and using statistical methods and data mining technique, we propose designing and implementing an automated system to generate peptide sequence. Here is the source of information of sequence comparison strategies using thermodynamic properties, characterize and obtain distribution of properties and characteristics by activity, implement distribution comparison script, compare distribution at level 1 using a statistical test all versus all strategy, compare and do correlation analysis at the level 1 all versus all, generate results and tabulate in summary format, and analyze the results and determine a relationship comparison, generate comparison matrices. Finally, review and analyze the results to discuss them. The second strategy consists of the comparison of sequence by means of alignment. The steps are summarized in a script to align two sequences in the same family, obtain adjacency matrix and conservation distribution by activity, scripts to align one versus the rest between sequence of different activities, a script to compare distribution between groups, a script to analyze quartiles and conservation membership between sequence of different activities. Finally, generate and analyze results and discuss. Here, the comparison of sequence using coded representations, escape distance through sequence in an activity, distance script 1 versus the rest, obtaining distance distribution, modify alignment script to identifying distance relationship. There are relationships between different properties. For example, there is a clear correlation between the properties of drug delivery peptides and transit peptides. However, sensory peptides are the only one that presents only direct dependence relationship, which greatly favors the design of this type of peptides when comparing thermodynamic properties. Neurological activity peptides show significant similarities at the phylogenetic level with sensory peptides, followed by drug delivery and immunological peptides. Remarkable, this type of approach facilitates the design and implementation of binary classification models of activity, considering this the concept of kinship between the analyzed sequence. Notoriously, unlike phylogenetic analysis, the numerical comparison shows relationship between neurological peptides and propeptides, which was not observed in phylogenetic analysis. An agreement was maintained with drug delivery peptides In this way, it shows that the application of different methods to study the same problem facilitates the identification of different patterns that help to generate these rules. Using the recognized statistical rules, the classification system bases on phylogenetic approach and the numerical distance methods, we design a methodology to create sequence with neurological activities. First, we define the rules. Next, apply a deep generative model. We create sequence. Next, we evaluate the sequence using the defined rules and check if proposed sequence is neurological or not. The iterations continue until the stop conditions are met. Finish, we report the sequence and its characteristics. Finally, we design and implement different computational methods to define the rules that characterize the activities of a peptide. These rules are based on statistical, thermodynamics, phylogenetic, and numerical points of view. Additionally, we propose and design pipeline of peptides with neurological activity, allowing us to obtain 34 new peptides with said activity. Despite the great advance, it is necessary to generalize this rule to the different existing activities and develop systems that evaluate peptides with moon-like characteristics and how to detect them. Acknowledgments to David, Karina, Bruce, Alvaro, Mehdi Davari, and Frances Arnold. I remain attentive to your question. Thank you so much.